Okay, guys, today is going to be a little short. Let's see. I want to break this one up into smaller units. So the way that it originally was intended was mitosis and meiosis all in one go. I'm going to separate it. This one is just going to focus on mitosis. So remember, the critical difference between mitosis and meiosis is that mitosis is how we generate identical cells. So when you're making new skin cells, that's mitosis. Meiosis, meiosis, all right, that's when we're producing gametes. So like making sperm and eggs, that's meiosis. So the way that I like to remember it is mitosis is how I made my toes, and meiosis is how my parents made me. Okay. So mitosis, again, we want a cell identical to the original. Okay. And we're gonna be getting more. I might make a just specific video just for the differences between mitosis and meiosis. But for right now, let's just get through mitosis for your understanding. So remember PMAT, PMAT, prophase, metaphase, anaphase, and telophase or telophase. Some people pronounce it differently. I don't care how you pronounce it. All right, so these are the steps in order, but let's see what happens before all that. So this is the standard cell cycle. Okay, so remember, interphase are all the steps outside of mitosis and cytokinesis. A cell spends most of its time in interphase. Okay, so if anybody ever asks you what phase does the cell spend most of its time in, you're going to be like interphase. Okay. It's not prophase, it's not metaphase, it's not mitosis or meiosis. A cell is spending most of its time in this step right here, just getting ready. Okay. So in G1, that's when we're duplicating everything we need, except for the chromosomes. So the other cell is going to need a mitochondria. The other cell is going to need a Golgi apparatus. It's going to need all that stuff. So we're getting it ready for cell division. Okay. So again, during G1, we're duplicating everything except for the DNA, the chromosomes. Okay. This 46 is specifically referring to the human chromosome count. Not every living thing has 23 pairs of chromosomes like humans do. In fact, most things don't. Okay. So for example, if we were trying to duplicate certain types of primates, they have more chromosomes than we do. So they might have 30 pairs for a total of 60 chromosomes. And if they're duplicating their DNA, and the original copy has 60 chromosomes, how many chromosomes are they going to need before they divide? 120, that's right. Okay. So this 46, do not get confused. This 46 is strictly referring to the number of chromosomes in the human genome. Okay. We have 23 pairs of chromosomes, and a pair is 2. And 23 times 2, let's do a little bit of math. What does that get us? 46. Very good. Okay, so G1, we're duplicating everything we need except for the genetic information. S stands for the synthesis phase. We're duplicating our chromosomes. G2 is when we're double checking, making sure there's no errors. This is a very important process because if there's anything wrong here, it's going to ruin the whole operation. Imagine if during your development, uh, a lot of your heart cells were made wrong. That could be problematic. Okay, so this is why it's critical for the cell to double check if there are any errors. And if there are, the cell performs apoptosis where it just politely kills itself. Um, cells that are not partaking in the cell cycle, for example, a lot of your nervous tissue cells, so like your brain and your spinal cord and all those cells, they are not duplicating. They're in G0. They're just chilling. All right. They're not duplicating. They're not doing any, they're just doing their job. They don't got time for duplication and all that nonsense. They're here to work. So interphase consists of G1, S, and G2. Interphase happens in both mitosis and meiosis. Okay. We're just doing this specific example for mitosis right now. Okay. So again, interphase, you got G1, S, G2. We're growing, we're duplicating everything we need, except for the chromosomes during G1. During S, we're making copies of our chromosomes. G2, we're making sure everything is good before we get into the division, okay? So here we go. So during interphase, we are preparing for the division. All right. And so the DNA during this phase, it's all inside of the nucleus for our eukaryotic cell. This is all for the eukaryotic cell, okay? It's packaged into a structure called a chromosome. So remember those chromosomes, those X things, that's all DNA, the whole shebang, it's DNA. But it's condensed. Before DNA is condensed into chromosomes, it's a soupy mess like we see right here. All right, so it's during interphase that it all condenses into a chromosome. So here are actual slides of an animal cell and a plant cell, okay? So mitosis has 4 stages. Remember what we were talking, we have prophase, metaphase, anaphase, and telophase, PMAT, okay? P-M-A-T, PMAT. All right, remember that. So during the very first stage, prophase, the chromosomes are duplicated, they condense and shorten, okay? It's during prophase that the membrane around the nucleus begins to dissolve and disappear, okay? Because remember, if we're gonna get these guys lined up down the middle to get pulled to opposite ends, we kind of need that membrane around them to go away. So that's what's happening during prophase. This is during mitosis, the PMAT phase, okay? The membrane around the nucleus begins to dissolve and disappear. The chromosomes are all nice and condensed. The spindle fibers are appearing. This is what's happening during prophase. Okay, the mitotic spindle disappears. This membrane around it breaks down. All right, here's another picture just to show you what's happening. All right, we got our prophase, we got our nuclear envelope breaking down, we got our chromosomes. This is all during step 1 of mitosis. Okay, nothing's happened quite yet. It's just preparing. It's condensing the DNA, breaking down the nuclear envelope. Spindle fibers are starting to appear. It's when we get into metaphase is when things start getting silly. All right, metaphase is very easy to identify because the chromosomes are all lined up so pretty down the middle, okay, on the mitotic spindle. All right, so it's during metaphase that our chromosomes are lined up right in the middle during mitosis. All right, so prophase, we have our DNA condensing in the chromosomes. The nuclear envelope begins to break down, the mitotic spindle appears. Metaphase, we got our chromosomes lined up right in the middle. Okay? So you don't need to know every single thing that's going on in each of these. You just need to know that during metaphase, it's lined up right in the middle for us. Here's another picture. Ain't it pretty? I drew this myself. I'm joking. I don't have nearly that level of artistic talent. Okay? So metaphase, again, it's the easiest phase to identify. Anaphase, now this is where I noticed there was a lot of confusion. We saw a lot of confusion between anaphase, telophase, and cytokinesis. So let me just break it down for you right now. Anaphase is when they're starting to separate and move to opposite ends. They're in the process of moving to opposite ends. They haven't made their way completely over there yet. That's just simply the process where we're pulling them apart and making them go opposite ends of the cell. All right, so again, prophase, nuclear envelope, begins to dissolve, chromosomes condense, spindle fibers appear. Metaphase, we got our chromosomes lining up right down the middle. And anaphase is when we're starting to pull them to opposite ends of the cell, okay? Now, telophase is when the mitotic spindle disappears, the chromosomes have made their way to opposite ends of the cell, and we got two new nuclear envelopes beginning to form. And see how the cell is starting to divide. We call that the cleavage furrow, this part right here where the cells are kind of going to want to pinch off from each other. Okay? So again, going back over it in order. Prophase, our chromosomes are condensed, our nuclear envelope is breaking down, and spindle fibers appear. Metaphase, our chromosomes line up right down the middle. Anaphase, they're starting to pull apart to opposite ends of the cell. Telephase, they're at opposite ends, a new nuclear envelope starts to appear, okay? Some of your texts just lump telophase and cytokinesis together. I personally prefer to make the distinction, but do not be surprised if you see, whether it be in certain textbooks or tests that just lump together cytokinesis and telophase. Again, I will always make the distinction, but not everybody does. So the membrane pinches in to divide the nucleocytoplasm organelles to form two daughter cells. Remember, these are identical to each other if everything went well. For mitosis, mitosis ideally wants to result in two identical cells to the original. Okay, that's the whole point. We're trying to make a cell that can do the same thing that this guy was doing. All right, so that is mitosis, the whole shebang. It's pretty simple. You just have to, shut up, gardens. You just have to, Hulu. Okay, so it's pretty simple. Try not to Google mitosis outside of what you're learning in here, because it gets a lot more complicated, and I promise you, I'm teaching you exactly what you need to know. Other places that you go to will try to tell you about phases between these things and the cyclins and all that. You don't need to know all that. All right. Everything I've told you about mitosis is all the district expects you to know about it. If you want to look deeper into it, I can give you the resources, but just try to stick to what we've gone over in here. Okay. So again, mitosis resulting in two identical daughter cells. Hemat are the steps to it: prophase, metaphase, anaphase, and telophase. Prophase, nuclear envelope breaks down, chromosomes condense, spindle fiber appears. Metaphase, stuff lines up right in the middle. Anaphase, start the chromosomes get yanked apart, pulled to opposite ends. Telephase, they're at opposite ends now, and the new nuclear envelope begins to form. And then cytokinesis, they pinch, and we just go back to interphase. Okay, so that is mitosis. All right, I'll make a separate video for meiosis, and then I'll make an additional video with the major distinctions between the two. Okay, but yeah, just try your best. Don't stress out. Yeah, I'm very good at ending videos. If you can't tell. Have a good day, or dull. It doesn't matter. Well, try to have a good day if you can. Don't force yourself.